Imagine a revolutionary new world of medicine where we can tackle diseases at the root cause, not just manage symptoms. That's the promise of CRISPR, clustered regularly interspaced short palindromic repeats, a game-changing gene editing tool that's like having a tiny pair of scissors for your DNA. CRISPR Cas9, to be precise, is the powerhouse combo. CRISPR is the DNA guide, and Cas9 is the enzyme that acts like molecular scissors, cutting DNA at specific locations. In this episode, we will be joined by a leading expert, Dr. Panos Chrysanthopoulos, to dive deep into the exciting world of CRISPR and its partner in crime, iPSCs-induced pluripotent stem cells. We will break down the science in easy-to-understand terms. You will learn how CRISPR-Cas9 can be used to create custom therapies for a vast array of diseases, from cystic fibrosis to cancer. We will explore how these therapies go beyond just treating the illness, but potentially curing it altogether. Forget medications with side effects. CRISPR-Cas9 gene editing and CAR T cell therapy, which is an abbreviation for chimeric antigen receptor using iPSCs, a personalized medicine on a whole new level. We will talk about the incredible potential of this approach and why it is a giant leap forward compared to conventional medicine. But here's the thing, not all medicines are created equal. Quality by design, or QBD, is a revolutionary approach to guarantee the safety and effectiveness of these new therapies. We will explain how quality by design ensures medication works exactly as intended. You will see why quality by design isn't just for big pharmaceutical companies, but holds the key to the future of safe and reliable gene-based therapies for everyone. So get ready for a tour of the future of medicine. We will be dissecting the science, exploring the possibilities, uncovering why CRISPR-Cas9 and quality by design are the dream team for a healthier tomorrow. Welcome back, dear listeners, to another thought-provoking episode of the Global Greek Influence podcast. I'm your hostess, Panayota Pimenidou, and you can subscribe on Spotify, Amazon Music, Apple Podcasts, and any of your favorite podcasting platform to never miss an episode as we bring together technology through scientific research, innovation, the economy, geopolitics, entrepreneurship, and yes, history, since they define and address our current and future challenges on Earth and possibly in space. Inspiring views, uncharted perspectives of global Greeks that make the difference. As we dive deeper into the ever-evolving landscape of science and technology, today's episode tackles a revolution brewing at the very core of medicine, genetic editing. Get ready, because we are about to explore the fascinating world of CRISPR, iPSCs, and how they're paving the way for a future of personalized therapies. Now, to guide us through this exciting frontier of medicine, I'm thrilled to welcome Dr. Panos Chrysanthopoulos. He is the CDO for Morphocell Technologies, a Montreal-based company at the forefront of cell therapies. Morphocell is pioneering on iPSC-derived liver replacement therapy, a potential lifesaver for millions battling acute and chronic liver failure. But Dr. Chrysanthopoulos' contributions extend far beyond Morphocell. He is also the co-founder and director of a cell therapist consulting group, helping other biotech companies across the globe leverage this transformative technology. Despite his impressive credentials, Dr. Chrysanthopoulos considers himself at his core a systems biology scientist, a testament to his deep passion for understanding life at the cellular level. Welcome to the show, Pat.
And thank you for having me over.
I'm really happy that you're here with us today, and let's get ready for our discussion. CRISPR is a groundbreaking technology with the potential to revolutionize medicine, as I've repeated many times. Just in December 2023, approved CRISPR therapies for sickle cell anemia have been approved by the FDA in the United States. However, with such power comes responsibility. So let's explore such a critical aspect. CRISPR, either ex vivo or in vivo, raises some ethical questions alongside its immense potential. How can we ensure responsible development and use of this powerful gene editing technology?
Yeah, that's a great question you're raising, Yota. And in my view, at least, CRISPR, although it does trigger these questions. These questions are not new. Any development in the history of science and technologies, any leap that we have demonstrated in those developments, we see that they raise similar questions. Now, CRISPR is a new tool to gene editing, but in reality, the ethical question is focused on the actual principles that CRISPR supports, which are the gene editing front of it. Therefore, this question is up for a discussion for the past 20, 30 years or so, because we have been genetically engineering cells to produce therapeutics for us for decades. We have been doing that with plants in order to generate phenotypes that are more robust and they can produce more of what we are using from the agricultural production and so on. So like in every advancement we experience, we see these spikes of ethical concerns. Now, I think it's also important to not only recognize the ethical questions, but also moderate a bit our responses to these ethical questions. And now to return to your original question, of course, what can we do about ensuring that these technologies do not get out of hand and we use them respectfully? I think that falls on the scientific community. We are 100% responsible on ensuring that everything we do, everything we discover has the single goal, and that is to advance humanity. Now, can we ensure 100% that these tools, everything that we're discovering in this life, will be used based on what the scientists had in mind during this discovery? The answer is no. Unfortunately, we have historical incidents where a technological advancement that can be used to advance humanity has been used for other causes or other purposes. So the intent behind the discovery is important. We need to ensure that whatever we're looking into as scientists, we're looking at under the prism of advancing humanity. But once our discoveries leave our hands, leave the labs, leave the universities and the companies, then we have a collective responsibility as humanity to embrace these discoveries and ensure that we apply checks and controls appropriately. That falls, of course, on government's regulations. It falls on public opinions. We have the responsibility of remaining educated, for example. What you're doing here with your podcast is a brilliant example of how society should respond to any technological advancement. So trying to break down the complex concepts that we deal with as scientists in order to be digestible by everyone, regardless of the educational background, is a very important first step to bring the society to a level where we can all create a common public opinion that focuses on controlling new discoveries and ensuring that their implementation is only for the betterment of our societies rather than just developing profits for different companies or whatnot. So I see there is a responsibility on the discoverer, the scientists. Then there is a responsibility on the facilitator, which could be the company slash university, and then the responsibility is also shared by the government with the regulations that they implement, and of course, in extension to that, by the public, the general public, who, of course, first of all, pick the government based on the mandate that they're promoting, and also they choose how to utilize the scientific advancements. I get it. That was a very long response to your question, but I somehow wanted to warm up a little bit the way we think about CRISPR and the ethical questions that you raised.
To extend a little bit this long question with the answer that you consider to be long, as you said, a groundbreaking discovery, regardless of what field this discovery might have been made, can get out of hand for revenue, profits, and so on. So I was wondering, since CRISPR has the potential to edit genes that influence intelligence, appearance, or athletic ability, where do we draw the line? For instance, one might say in 10 years' time, I want to be as smart as Panos. I want to be as athletic as Panos. What's happening in this case?
That is a good question. And in reality, the levels that somebody needs to go through in order to apply any discovery, any novel discovery on a human are very complex, and there are multiple layers of approval that anything that we do has to go through before it reaches any human. So there is a big leap that somebody needs to take conceptually in order to think the potential of somebody using CRISPR in order to enhance characteristics of an adult. Again, think about it. When we have a therapeutic, in order for the therapeutic to go into the clinic, you need to demonstrate, first of all, first and foremost, its safety through preclinical studies, through animal studies. You have to demonstrate that your therapeutic is safe, and you have to demonstrate that your therapeutic has an impact on a condition, on a disease, in order for you to get any approval to take your discovery and apply it in the phase one clinical trials. So that's the first barrier there, that you cannot go through. You cannot apply anything on any human unless you get approval from the regulatory bodies. Now, the good news for us as humanity is that the global regulatory bodies across all jurisdictions, they want to work together. They want to come together. They want to harmonize the requirements for any study to go into the clinic. So we see now that there is a global umbrella forming of regulators in North America, in Europe, in Japan, that come together, and they started establishing a framework of regulations or guidelines regarding what kind of prototypes, what kind of therapeutics, of applications of our discoveries can actually go through the clinical trials and be applied on humans. So no matter how crazy I might be as a scientist and I want to try whatever it comes to my mind on humans, the reality is that in real life you can't. There is so many layers that you need to go through that it's practically impossible to do so. Secondly, in order to develop and apply any of these discoveries, it actually takes a lot of money. The investment is huge. Therefore, even if somebody wanted, if a single individual was crazy enough and they found a loophole to go through this first layer of control, they would still need to be backed by serious money. And as we know, any money flow, any transactions of big amounts of money attracts attention, attracts other regulators of the government, and therefore it's an extra layer of control during that phase one. Even if a therapeutic goes into a phase one, let's assume that there is some sort of a weird application of a new discovery that is questionable ethically and goes through phase one, then again, the interaction with the regulators are so frequent and the diligence that the regulators perform are so thorough that it's extremely hard for anyone to go through with a questionable application of any discovery. So discussing about these potential applications is a great philosophical discussion, and it can push us to explore certain ethical questions. But I think in reality, we should be assured, we should feel calm as a society that there is multiple layers of control, multiple layers of regulations that any therapeutic, any application, scientific application needs to go through in order to go and be applied on a human. To give you an idea, we are developing a liver replacement therapy within Morphocell Technologies. And for us to convince the regulators that our therapy is safe and efficacious to be tested on humans, it's a process that will take us 3 years and many millions of dollars in order to collect all the data that will convince the regulators to allow us test our therapy on a human. So I hope that what I just described provides a little bit of calmness to the broader audience, especially after the COVID era. I totally understand that the emotion is quite high and the uncertainty exists, mainly triggered by some irrational decisions that were made by certain governments all around the globe. But what I want to reassure the people on is that the path of any discovery to get to humans is a very, very structured path, and it's extremely tough for anyone to convince the regulators to allow therapeutic testing on humans.
Some crucial points, Pano. We have talked about ensuring responsible development of CRISPR, but ensuring equitable access is equally important. Right now, many cutting-edge treatments are out of reach for many patients. Will CRISPR therapies be different?
CRISPR therapies can be divided in two categories. So CRISPR is the technology, the tool that we use to perform a very precise gene edit. There's a number of diseases out there that are caused by point mutations. So there are humans that carry a point mutation that causes severe and sometimes lethal diseases. With the introduction of CRISPR, of in vivo application of CRISPR, the application of the tool on a living human, we are able to actually revert these diseases and give back to these patients a normal life. Although the therapy, the in vivo application of CRISPR at the moment is still very, very expensive, if you put it on the perspective of the whole lifespan of an individual that is suffering from one of these severe diseases, CRISPR gene editing, it's actually affordable even today in its in vivo application. Now, like every technological advancement, these are the first steps that we're taking. We are making the baby steps. The field is very young, and we're expecting that the cost of application, the cost of goods, the cost of manufacturing of these things, the cost of controlling and monitoring after the application of these therapies is still very expensive. However, the reality is that as we progress with the iterations and through a broader application, these therapies will become affordable as we move forward. The second application of CRISPR, which is the most expensive actually application at the moment, has to do with a CRISPR-based editing of cells of ex vivo. So we're isolating cells from a patient or a healthy donor, and then we submit those cells to a gene editing in order to give those cells specific attributes that we want them to have in vivo once they really get reinjected. Now, that arm of therapies that is affected by CRISPR is a field where we can definitely make important and brave advancements on cutting costs. We can do that by the industrialization of manufacturing of these processes. We can do it by introducing automation. We can do it by scaling up manufacturing and ensuring that these therapies actually reach the patients that they so much need. Yes, like everything, like computers when they were first introduced in our society, cars, any discovery in our history was extremely expensive up front, and through iterations, through time, these things become more trivial by the hour, I could say. If you think about how much it would cost to sequence a whole genome a decade or 15 years ago and how much it costs today, it's night and day, right? It's today's at an absolute trivial amount. Back 10 years ago, that was an exuberant amount of money that you would require to sequence a gene. So things progress. I'm very optimistic that these therapies will be broadly available to everyone.
Absolutely. Even in the renewable energy field, engineers hope and they know, because it's been a part of our collective experience, that as we scale up a process or a system or a new technology, it becomes cheaper because it becomes more mature. We most often refer to it as learn by doing. So we know that as we learn to do things more often at a greater scale, then these technologies become more affordable and more accessible. Pano, you mentioned the importance of responsible development in CRISPR therapies. One key aspect of responsible development is ensuring these therapies work effectively for a wide range of patients. Interestingly, there is a whole approach in medicine dedicated to this very issue. It is called quality by design, or QBD. Why some medications seem to work differently for different people? QBD is trying to solve this puzzle. It might seem unrelated to cutting-edge gene editing, but quality by design could play a pivotal role in ensuring the safety and efficacy, including future CRISPR therapies. So, diving deeper, how can quality by design help us ensure these new therapies work for everyone?
Such a loaded question, Yoda, because the reality is that the different impact that we see that these therapies have on a population is affected by multiple factors. So first of all is the obvious, which has to do with the variability of the actual manufacturing of these therapies. These therapies are very, very complex. They require highly skilled and highly trained individuals to manufacture these therapies, and they are very manually involved at the moment. Because we're new, because we haven't automated yet these approaches, because we're still discovering, we're still learning, this unfortunately leads to a more variable manufacturing process. Therefore, the products end being a little bit on the more variable end of the spectrum. Now, this is an area where QBD can really help, but we'll return to this because there's other elements that we don't usually consider that contribute towards this variable outcome of the therapies. And those have to do with, first of all, the way we classify diseases. As a medical community, have unfortunately or fortunately, we are forced to push multiple adjacent conditions that demonstrate similar symptoms. We're forced to push them under the umbrella of a single disease. So the reality, though, is that a person that has a melanoma, one type of skin cancer, when compared to another person that has a melanoma, again, skin cancer, which in theory fall under the same category, the same disease, the reality is that they demonstrate a very different phenotype, a very different cluster of symptoms. So they may have the core disease symptoms appear to overlap, but in reality, the full spectrum of characteristics are different. So the patient population variability is also a very important reason why we see these therapies perform differently over the population. Now, go back to the QBD aspect of it and how QBD can help us reduce this variability and ensure that the highest possible quality is introduced into our product. I would like to give a little bit of an intro as to what QBD is. What does it stand for? It's a framework of thought that forces the product developer to introduce quality into the product. You do so by building early on a deep understanding of how your manufacturing process works. And what that means is that when you manufacture something, there is a lot of moving parts, there's a lot of variables, there's a lot of things that change. If you build an early on deep understanding on how these changes impact the attributes of your product on the back end, if you understand fully that, if you move the item A from here to here, what is the exact impact that this movement would have on the qualities of your product? QBD forces you to break your system apart and ensure that you understand these things. You understand that if you had a deviation during manufacturing, if somebody took, instead of 30 minutes, took an hour to put the cells in the freezer, for example, QBD forces you to understand what's the exact impact that this deviation will have in the quality of the product at the back end. Therefore, the manufacturers, the product developers, they understand fully what are the changes that are important and what are the changes that don't really matter on the quality of the product. So indeed, QBD is a framework of thought that has been applied, by the way, in the different field of biologics for decades now. It's not something new. The application, though, in our field is very new, and I'm personally very excited to see it being more discussed, being more of a topic, because I strongly believe that for therapeutics such as cell and gene therapies, which are very, very complicated products, the deployment of a framework of thought such as QBD is extremely important because we need that deep understanding of our complex products. We need to know how these products behave in vitro as well as in vivo. So I strongly believe that QBD can have a serious impact on ensuring similar behavior, a similar performance of these therapeutics on a broader population.
One understands that QBD for CRISPR therapies are more challenging than quality by design for 100 milligrams of paracetamol tablets, just because CRISPR therapies are personalized therapies. So just imagine the complexity of QBD for CRISPR therapies. A key player in this exciting field of gene editing is the use of induced pluripotent stem cell or iPSCs. This might sound complex, but let's break it down. Imagine having a cell with a remarkable ability to transform into any cell in the human body. That's essentially what an iPSC is. Scientists can take a regular adult cell, like a skin cell, and reprogram it back to an embryonic-like state using special factors. These reprogrammed cells, the iPSCs, then have the incredible potential to become any cell type we need, including the kind of immune cells used in CAR T cell therapy. This is particularly exciting because it allows us to create personalized therapies. Now, Pano, can you elaborate on how exactly iPSC-derived cells are generated and what advantages and limitations come with using them in CAR T cell therapy?
Of course, of course. You very well described it, that iPSCs are just cells that we have isolated from a donor and we have reprogrammed to demonstrate this pluripotency. Now, these cells are very similar to what the phenotype of an embryonic stem cell looks like. And embryonic stem cells are the cells that we have when the embryo is first started developing. So these cells in the embryo, throughout the development of an embryo, get different signals. And these signals are just proteins or chemicals from their environment. Depending on the amount, depending on the nature of those signals, depending on the duration of those signals, these cells decide their fate. So think about it, we start from a single cell, from two cells, and we become the complex organism we are. The cues for these cells to become something different, to differentiate into a different tissue, a different cell type, is through their environment. So what we're doing in the lab is we take these iPSCs and we try to mimic what happens on the embryo during the differentiation of different tissues. So we have managed to identify protocols and methods that drive our iPSCs down any type of lineage, any type of a cell, of a mature human cell on the body, by just playing around, manipulating the types of signals that we provide to these iPSCs, the duration and the concentration of these signals. Through Morphocell Technologies, for example, in our case, we drive iPSCs down the path of a hepatocyte. So we create hepatic cells through this type of manipulation from iPSCs. Now, this approach has a tremendous advantage on scalability. And why is that? Fully differentiated cells on our human body rarely have the capacity of proliferate, of growing. They're usually slow at growing and regenerate. However, iPSCs are fast growing cells. They double in every 20 hours, 24 hours, they double in numbers. So when somebody wants to develop a therapeutic to regenerate certain tissues on a larger population, you need to somehow build that scale. You know it very well. Your chemical engineering background allows you to understand how important scalability in manufacturing is. So with iPSCs, what we can do is scale the manufacturing, the process in the early stages of the manufacturing while our cells are still iPSCs and they grow fast. So we build a big bank of them, and then we take parts of the iPSCs and we drive them down the lineage that we want based on the therapeutic we want to develop. So it allows us to develop cheaper, more affordable, and more accessible medicine to the public. A second thing that they allow us as iPSCs is that we can develop off-the-shelf therapies. We can develop therapies that are universal. One thing that we need to keep in mind about cell therapies is that, like transplants, if somebody gets a cell therapy that is developed from cells of another donor, your immune system will reject this therapy. Your immune system will respond to it. If we want to start looking into these off-the-shelf therapies, we need to go back to the iPSCs and try and genetically engineer, using CRISPR, genetically engineer these iPSCs to remove the proteins that trigger the immune system of the host. So what we're currently doing as a whole community is trying to identify the exact combination and the methods to knock out a specific subgroup of genes and allow these iPSC-derived therapies to be invisible to the immune system of anyone. Therefore, if anyone gets treated with this therapy, they don't need to be immunosuppressed. They can have a very normal life, which is fantastic, and it can only be done because we have the iPSCs, because we have this central bank where we can engineer a small amount of them, grow them in very large numbers, and then differentiate them to their therapies. As you understand, these two things that I brought up have a serious impact on the cost of developing these therapies and supplying these therapies to the public. Therefore, it's a no-brainer that iPSCs have a very serious impact on availability of cell therapies to the broader public. Now, like everything in life, iPSCs don't come without disadvantages. As I mentioned earlier, if one doesn't engineer the iPSCs, the iPSC-derived cell therapies will trigger the immune system. So we cannot just use iPSCs that we have reprogrammed from a cell type and just apply it on any patient, because their immune system will reject it. So we need CRISPR. We need to edit them before we apply it on any patients. And then the other concern that iPSCs raise has to do with what I described as an advantage earlier. So it's an advantage and a disadvantage. And what that is the potential to proliferate, to just continuously propagate. That raises a concern, because if an iPSC is not differentiated, and it remains as an iPSC, that cell has the ability to grow. And they grow in a sphere formation. We're talking about cells that have the potential to develop a tumor in the host. Of course, these are all risks that the developers know early on, and these are the risks that the regulators around the world are always focusing on and drilling all the developers in order to develop proper analytics and ensure that this risk is properly mitigated. Last but not least, the big disadvantage of the iPSC-derived cell therapies is that the whole process of reprogramming them, banking them, and then differentiating them in any tissue that we need currently requires a very, very well-trained expert operators. That, of course, makes the therapies expensive at the moment. Until we get to a point where we can industrialize these processes, I'm afraid that given the nature of the product, iPSC-derived therapies are still on the expensive side.
In CAR T cell therapy, T cells are removed from a patient's blood. These T cells are then modified in the lab to include a special receptor called a chimeric antigen receptor, CAR. The CAR helps the T cells recognize and attack cancer cells. The modified T cells are then infused back to the patient's blood. The CAR T cells then multiply and attack the cancer cells. CAR T cell therapy is a type of cell and genes therapy, CNGT. What diseases do you see CNGT therapies having the biggest impact on in the next 5 to 10 years?
As I alluded to earlier, CRISPR-based can have a very, very immediate impact on conditions and diseases that are caused by single point mutation. This is something that, as you mentioned in the beginning of our discussion, I think you mentioned the sickle cell disease, that it's their single point mutation diseases will be, in my view, in my opinion, in the next 5 to 10 years will become obsolete. We will have a permanent solution for such diseases. That's my projection, my perspective. And I think that that's the area that cell and gene therapies will have the most impact immediately, right?
Can you give us a few examples of these single cell diseases that can be erased from humanity forever in the next 5 to 10 years?
For us, coming from Greece, we're very familiar with thalassemia, anemia. There is a lot of blood-related conditions that are single point mutation derived, thalassemia, sickle cell disease, hemophilia, and also Huntington's disease is another single point mutation derived disease, and a lot of others that my brain doesn't help me recalling all of them on the top of my head, but there's a series of very, very serious diseases, and those patients' lives can change overnight through these therapies. Having said that, as you very well mentioned, CAR T therapies have already made a small revolution in blood cancer therapies. I think that blood cancers will also become obsolete quite soon. Now, the reason why we're separating blood cancers from every other cancer is because they're soft tissue cancers. And one of the trickiest things that we're still trying to solve in CAR T therapies is how can a CAR T cell penetrate a tumor, a solid tumor. In the bloodstream, where we're talking about leukemias, they don't have this barrier. They don't need to enter a hypoxic environment of dense tissue to try and find the core of the tumor. They can find the periphery, but penetration into the tumor is a tricky part. So blood cancers, in my opinion, will come first. And then I'm pretty sure we're going to solve the issue of penetration in solid tumors. And at the end, I think that we've already started seeing great advancements in tissue regeneration. A former employer of mine, Blue Rock Therapeutics, is already in the clinic with a dopaminergic neuron replacement therapy that fights Parkinson's disease. Especially when we look at the brain, neural degeneration is something that we will have to deal with quite soon as humanity. We are aging as global population, and unfortunately, our brains don't age as well as our bodies do. So fighting this type of diseases is something that we are focusing on, and it's something that we see immediate results on. At the same time, liver disease is what I'm currently tackling through Morphocell Technologies. We are developing liver replacements to fight liver failure. We are actively, as a field, we're in the clinic with multiple approaches to replacement of pancreatic cells for fighting diabetes and so on. So single point mutation, cancers, oncology overall, and tissue regeneration is the areas that I strongly believe that in the next 5 to 10 years, cell and gene therapies will have a revolutionary impact.
We have been discussing the incredible potential of new therapies like CRISPR and CNGT, but sometimes even the most promising treatments can face setbacks. One major concern is medication recalls. This can happen for various reasons, but often it is because the medication's effectiveness or safety wasn't consistent during production. This is where the concept of quality by design, or QBD, comes in, and which we have touched upon. It might sound technical, but QBD is essentially a proactive approach to ensure medications are manufactured consistently as safety right from the design stage. And you've also described all the concerns and considerations that need to be taken into account during QBD. To explore this further, Pano, we have heard of medications being recalled due to safety or efficacy issues. How can quality by design help prevent this situation? If you could think of any extra points relevant to the later stages of some therapist production.
I think the implementation of QBD framework has the ability of reducing the number of recalls. It can achieve that by ensuring a robust manufacturing process, a process that we understand whether it's going to fail or not in the quality of the final product, because as I described earlier, QBD forces you to build a deep understanding of the manufacturing processes. Now, that's the main way that you can ensure the quality of the product at the back end. At the same time, it forces you to build proper controls in order to ensure that for critical parameters, your manufacturing process does not go beyond the failure mode, beyond the failure thresholds. So that's another way that it forces you to develop something more robust. And of course, as I mentioned, understanding the process, understanding your analytics, ensuring proper controls makes for a far more reproducible product manufacturing on the back end. Having said that, though, although QBD can reduce and has the ability to reduce the recalls of medication due to quality issues, when it comes to therapeutics that are recalled for side effects that were not known before, this is something that, fortunately or unfortunately, as a society, we have to deal with. And the reality is that we as humans are so complex as organisms that it's impossible for anyone to know exactly the impact that every therapeutic can have on every single individual on the planet. The clinical phases of development try to ensure that we get a representative sample of the population and test our therapeutics on that representative sample. Now, that doesn't mean, of course, that that representative sample is always enough for us to ensure 100% that these therapeutics will not have any other side effects that have not been discovered yet. Now, I'm not saying this to scare anyone by any means of imagination, right? We do our best. And the reality is that we have to strike the right balance, because if we waited for us to be 100% sure that our therapies do not have any other side effects on the population, we will not have any therapies in the market. That's the reality. Unfortunately, we're trying to strike the right balance. We're trying to save lives of people that are suffering day in, day out, while at the same time ensure safety on the population level. But we will always see some events, other events of medication recalls. And again, I insist and I emphasize of the rare. And I'm saying that because we pay attention as a human brain by instinct. We have focus on identifying the danger. We focus on identifying what is bad for us. That's our surviving instinct. Therefore, we tend to focus more on the recalls of the medication rather than the deployment of medications. So in order for us to feel a bit calmer about these recalls, all we have to do is just go online and figure out how many drugs are actually released every year, right? And how many of those get recalled for safety issues. The fraction of that is so small that I think every individual out there can really understand that this is not a serious concern.
QBD sounds like a powerful tool, but this might be our last question for Dr. Chrysanthopoulos today. The good news is the principles of QBD can be incredibly valuable to smaller companies as well. By focusing on understanding the manufacturing process and building quality into the design phase, even smaller companies can benefit from greater consistency, efficiency, and ultimately safer medications for patients. So, Pano, can smaller companies with limited resources effectively adopt quality by design principles? If so, what are some practical tips for them to get started on this journey?
Fantastic question. I love this question because this is an uphill battle for me in my career. Because there is this misconceived notion about the cost and resource investment that goes into QBD deployment. The reality, though, is that QBD is better suited for smaller companies, for companies that cannot afford having any waste, for companies that need to be very focused and need to ensure that every step they take. QBD, because it's a risk-based systematic framework of thought in product development, it allows the small companies to focus on what really matters. So they can very strategically deploy their limited resources on things that actually have an impact, on things that have moved the needle, as we say. First. Second, small companies that do not have the proper infrastructure of software and quality management systems and all these tools that are necessary to record and permanently store information in a structured manner. QBD allows the small companies that don't have all the infrastructure create a kind of a memory, historic memory of the reasons why they made the decisions that they did and what were the data that informed certain decisions during the development of the product. So I strongly suggest to any company out there to really engage and really try to deploy QBD. My advice to them in order to deploy properly is to try and identify somebody that can help them in the first steps, somebody that can build some basic infrastructure and some basic understanding on a few SMEs within the company, so it can then be deployed properly. There is a lot of consultants out there that can help with it, including myself and my partners, but there's a lot of people that understand and know the principles of QBD, and they can support smaller as well as bigger companies to deploy it. But I couldn't recommend it more for any company that is willing to move ahead in an efficient manner.
Wow, Pano, that was a mind-blowing exploration of the future of medicine. We covered so much ground today, from the revolutionary potential of CRISPR gene editing and the power of iPSCs to the personalized approach of CAR T-cell therapy and the importance of quality by design for ensuring safe and effective medications. It's a truly exciting time in medical science, and you've given our listeners a fantastic glimpse into the possibilities that lie ahead.
Thank you for having me, Yota. It's always a privilege trying to connect with the public. I hope I did not confuse anyone, any of your listeners, and I wish you all the best. And I just want to say a big thank you for not only inviting me, but also for the service that you're providing to your listeners.
Once again, so much, Pano. A huge thank you for joining us on this exciting journey into the world of gene editing and personalized medicine. We hope you are as inspired as we are by the potential of these technologies to revolutionize healthcare. Stay tuned. We've got even more fascinating topics and interviews lined up for future episodes. And don't forget to connect with us on social media platforms like X, Facebook, and LinkedIn. We love hearing your feedback and suggestions for future guests, or maybe even if you would like to be a guest yourself. Thank you once again, Dr. Chrysanthopoulos, Pano, and thank you all of you for listening.